Imagine your brain as a pristine, high-functioning city. For decades, the traffic flows smoothly, the waste management systems are efficient, and the power grid is stable. But then, slowly, almost imperceptibly at first, the garbage trucks stop running. Debris begins to pile up in the streets. This debris isn't just litter, it is toxic, sticky, and indestructible. It clogs the intersections, causing traffic jams. It shorts out the power lines, causing blackouts. Eventually, the city shuts down. This is the reality of Alzheimer's disease. For over a century, since Dr. Alois Alzheimer first peered through his microscope in 1906, we have identified this garbage as beta-amyloid plaques and tau tangles. And for decades, the entire might of the pharmaceutical industry has been focused on sending crews into the city, into the brain, to scrub these plaques away. We have developed antibodies, vaccines, and secretase inhibitors, yet the city often continues to crumble. But what if we have been looking at the problem from the wrong angle? What if instead of sending cleaners into the brain, we could simply lower the pressure outside the city, creating a vacuum that sucks the garbage out naturally? Right now, a groundbreaking exploratory clinical trial is testing a therapy that doesn't target the brain directly at all. Instead, it targets the blood. By utilizing a highly advanced, lab-grown version of the most abundant protein in your plasma, Recombinant Human Serum Albumin, researchers are attempting to turn the patient's bloodstream into a peripheral sink, a mechanism designed to drain the toxic amyloid out of the brain and into the blood, where it can be safely eliminated. This is not science fiction. This is the Peripheral Sink Hypothesis in action. And if this trial succeeds, it could revolutionize how we treat neurodegeneration, moving us away from invasive brain treatments towards a scalable, blood-based solution. Welcome to the deep dive. Hello, and welcome back to Clinical Trials Explained, the channel where we bridge the gap between complex clinical data and the human experience. I am your host, and today we are embarking on a significant journey into the heart of neurodegeneration science. We are not just skimming the surface today. We are going to deconstruct a specific, active clinical trial, piece by piece. We will explore the molecular biology of the drug involved, the historical context of the disease it aims to treat, the intricate design of the study, and the immense ethical and safety considerations that surround it. Our focus is a study titled Recombinant Human Serum Albumin in the Treatment of Alzheimer's Disease. It is an exploratory trial, a critical first step in determining whether a synthetic protein can arrest the decline of the human mind. Before we begin, a necessary disclaimer. The content presented here is strictly for educational and informational purposes. I am a narrator analyzing public scientific data, not a doctor. This is not medical advice, nor is it a recommendation to join any specific trial or take any specific medication. Clinical research is experimental by nature. Outcomes are never guaranteed, and risks are always present. Now, settle in. We have a lot of ground to cover. To understand where this trial fits in the grand tapestry of medicine, we first need to understand the magnitude of the enemy it faces. Why does this specific trial matter? To answer that, we must look at the landscape of Alzheimer's disease today. It is often called the silver tsunami, a demographic inevitability that threatens to overwhelm healthcare systems globally. Alzheimer's is not merely a disease of forgetting keys or losing names. It is a progressive, fatal, neurodegenerative condition. It physically destroys the brain. If you were to look at a scan of a patient in the late stages of Alzheimer's, you would see significant atrophy, shrinkage. The hippocampus, the seat of memory, withers away. The ventricles, the fluid-filled cavities in the brain, enlarge to fill the void left by dying neurons. Current treatments are limited. For decades, we relied on cholinesterase inhibitors like donepezil, which treat symptoms but do nothing to stop the dying of cells. Recently, we have entered the era of anti-amyloid monoclonal antibodies, drugs like lecanemab and donanemab. These are major scientific achievements. They prove we can remove plaque from the brain. However, they come with caveats. They require intravenous infusions, they are incredibly expensive, and they carry the risk of ARIA, amyloid-related imaging abnormalities, which involve swelling or bleeding in the brain. This brings us to the unique importance of this trial. This trial investigates a different approach using albumin. Albumin is the workhorse of your blood. It constitutes about 50 to 60% of all the proteins in your blood plasma. Its job is transport. It grabs onto hormones, fatty acids, and medications, ferrying them around the body. Crucially, it also binds to toxins, including beta-amyloid. There is a theory in medical science called the Peripheral Sink Hypothesis. The idea is based on equilibrium. Your body strives for balance. If the concentration of soluble amyloid is high in the brain and low in the blood, the laws of physics dictate that the amyloid should move from the high concentration area, the brain, to the low concentration area, the blood, to balance things out. However, in Alzheimer's patients, the albumin in their blood is often glycated or oxidized. It's damaged and dirty. It's already full of junk. It cannot bind to amyloid effectively. Therefore, the sink is clogged. The amyloid stays in the brain, clumping into plaques. Previous studies, most notably the AMBAR trial, tried to fix this by performing plasma exchange, literally taking a patient's blood out, separating the plasma, throwing it away, and replacing it with fresh plasma from healthy donors, and it showed promise. But plasma exchange is invasive. It requires big machines, central lines, and crucially, it relies on human donors. You need thousands of liters of donated blood to treat patients. It is not easily scalable to millions of people. That is why this trial is so critical. It uses recombinant human serum albumin, rHSA. Recombinant means it is not taken from blood donors. It is made in a lab, usually by genetically engineering yeast or bacteria to produce the human protein. If this trial shows that injecting lab-grown albumin can clean the brain just as well as a full plasma exchange, it solves the scalability problem. We wouldn't need millions of blood donors. We could manufacture the treatment in vats, just like we make insulin. This matters because it represents a shift from a scarcity-based treatment model to an abundance-based manufacturing model for treating the world's most costly disease. What researchers are studying? Let's get technical. What exactly is the substance being investigated in this trial? The intervention is recombinant human serum albumin, rHSA. To understand rHSA, we must first understand SHA, human serum albumin. Albumin is a single-chain polypeptide containing 585 amino acids. It is synthesized in the liver and exported into the bloodstream. It is a negatively charged protein, which allows it to act like a sponge for positively charged ions and various molecules. In the context of Alzheimer's, researchers are interested in albumin's high affinity for beta-amyloid peptides. Beta-amyloid is a sticky fragment of a larger protein called the amyloid precursor protein, APP. In a healthy brain, these fragments are broken down and cleared away. In Alzheimer's, they stick together, forming oligomers and eventually fibrils and plaques. The researchers in this trial are operating on the premise that administering fresh, high-quality recombinant albumin intravenously will increase the binding capacity of the patient's blood. Think of the blood-brain barrier, the defensive wall separating your brain tissues from your bloodstream. It is semi-permeable. Small molecules can cross, large ones usually cannot. However, there are transport mechanisms. Soluble beta-amyloid can cross this barrier. The hypothesis works like this. 1. Administration. The patient receives an infusion of rHSA. 2. Binding. This fresh albumin circulates in the blood. It is empty and hungry for things to bind to. 3. The gradient. Because the blood now has a high capacity to hold amyloid, the concentration of free amyloid in the blood drops effectively to 0 because it all gets bound to the albumin. 4. The pull. This creates a steep concentration gradient. The brain is full of free amyloid. The blood has none. Following the gradient, soluble amyloid flows out of the brain, across the blood-brain barrier, and into the bloodstream. 5. The trap. Once in the blood, the amyloid is instantly snapped up by the fresh albumin. It is trapped. It cannot go back into the brain. 6. Clearance. The albumin-amyloid complex is eventually processed by the liver or kidneys and excreted from the body. This study is specifically looking at mild to moderate Alzheimer's disease. This distinction is vital. In severe late-stage Alzheimer's, the damage to the neuronal architecture is catastrophic and likely irreversible. The neurons are dead. No amount of amyloid clearing will bring them back. However, in mild to moderate stages, the neurons are struggling, but many are still alive. The synaptic connections are fraying, but not severed. The logic is that you can reduce the toxic load now, you might salvage the remaining function and halt the progression. The researchers are also testing the safety of recombinant albumin. Historically, albumin used in hospitals comes from fractionation, processing pooled human plasma from thousands of donors. This carries theoretical risks of pathogen transmission, though modern screening is excellent, and supply chain limitations. Recombinant albumin eliminates the pathogen risk entirely, but it introduces a different concern: immunogenicity. Because it is made in a non-human host, like yeast, there is a tiny risk that the final product might contain trace amounts of yeast proteins, which could trigger an allergic reaction in the patient. This trial is rigorously monitoring for that. Now, let's dissect the protocol of this protocol. How do you prove that a protein injection can save memory? You need a rigorous, controlled environment. Study design. This is an open-label, parallel group, randomized exploratory study. Let's break down those terms, as they define the quality and nature of the evidence being generated. Exploratory. This means the study is in an early phase. They are likely looking for signals of efficacy and establishing safety protocols. They are not expecting to get FDA approval solely based on this one study. They are laying the groundwork for larger phase 3 trials. Randomized. Participants are assigned to groups by chance, like flipping a coin or using a computer algorithm. This minimizes selection bias. You don't want the doctor putting all the healthiest patients in the high-dose group, which would skew the results. Parallel group. This means the groups run at the same time. Group A gets one dose, Group B gets another, and they are compared side by side. Open label. This is a crucial detail. In an open-label study, both the researchers and the patients know what they are receiving. There is no placebo group in the traditional blinded sense here. While this introduces some bias, the placebo effect, it is often done in early-stage trials for safety reasons and ethical considerations, or when the logistics of a placebo infusion are difficult. However, it does mean we have to interpret the efficacy data with caution. The cohort. The trial aims to enroll 30 subjects. This is a small number, consistent with an exploratory Phase 1/2A trial. Inclusion criteria. The participants must meet the National Institute on Aging and Alzheimer's Association, NIAA criteria for probable AD dementia. They need to be in the mild to moderate stage. This is usually defined by a mini mental state examination, MMSE score, often between 10 and 26. Exclusion criteria. Safety is paramount. They exclude patients who have received plasma derivatives recently to avoid confusing the results, those with a history of organ transplantation who might have immune issues, and crucially, those with irreversible visual or auditory impairments. Why? Because if you can't see or hear well, you cannot complete the cognitive tests, and your data would be invalid. The arms and interventions. The participants are randomized in a 1-to-1-to-1 ratio into 3 groups. This is a classic dose-ranging design. They want to find the Goldilocks dose, effective enough to work, but not so high that it causes side effects. 1. Low-dose group receives 20 grams of recombinant human serum albumin. 2. Medium-dose group receives 30 grams of recombinant human serum albumin. 3. High-dose group receives 40 grams of recombinant human serum albumin. The study duration is roughly 1 year, with a primary completion date estimated around November 2025. The endpoints. What are they measuring? To determine if the drug works, researchers use endpoints. Safety, primary objective. First and foremost, do the patients tolerate the infusion? Do they develop fever, rashes, or fluid overload? Albumin pulls water into the blood vessels. If you pull too much, you can strain the heart. Volume overload. This is a critical safety metric they will watch closely. Efficacy, exploratory objectives. ADAS-COG, Alzheimer's Disease Assessment Scale, Cognitive Subscale. This is the gold standard in AD trials. It's a test that measures memory, language, orientation, and praxis, the ability to follow commands. It consists of 11 tasks. A score ranges from 0 to 70. Crucially, in ADAS-COG, a higher score means more impairment. So the researchers are hoping to see the scores stabilize or decrease, improve, in the albumin groups. NPI, Neuropsychiatric Inventory. Alzheimer's isn't just about memory, it's about behavior. Patients often suffer from agitation, anxiety, apathy, and hallucinations. The NPI measures these symptoms. A lower score indicates better behavioral health. This is vital for caregivers, as behavioral issues are often the reason patients are moved to nursing homes. ADCS-ADL, Activities of Daily Living. Can the patient dress themselves? Can they use a telephone? Can they prepare a meal? This score ranges from 0 to 78. Here, a higher score is better, more independence. The researchers will measure these scores at baseline, start of the trial, and then at weeks 7, 16, 25, 29, 37, and 41. This frequent monitoring allows them to plot a trajectory. Is the decline slowing down compared to historical data?
If, and it is a big if, the data from this trial is positive, the implications are staggering. Let's look at the logistics. Currently, the leading treatments, anti-amyloid antibodies, require complex monitoring for brain swelling, ARIA. They require MRI scans before and during treatment. They target the brain directly, which is a sensitive organ. If albumin therapy works, we are talking about a treatment that works centrally, on the brain, by acting peripherally, in the arm. This concept of systemic treatment for central disease could open doors for other conditions like Parkinson's or ALS. Furthermore, the recombinant aspect is the game changer for access. Natural human albumin is a finite resource. It relies on plasma donation centers. There are frequent shortages of albumin in hospitals, particularly in developing nations. If we rely on donor plasma to treat the millions of Alzheimer's patients, we would drain the global blood supply dry. Recombinant albumin is manufactured. It is scalable. A factory can produce tons of it. This means the cost could theoretically come down over time, making it accessible not just to the wealthy elite, but to the global population. However, we must temper our optimism with scientific realism. The Peripheral Sink Hypothesis is elegant, but it is not yet proven to cure Alzheimer's. Some critics argue that pulling amyloid out of the blood is like bailing water out of a sinking Titanic with a bucket. It helps, but if the hole in the hull, the underlying disease driver, is too big, the ship still sinks. Others argue that amyloid is the wrong target entirely, or that by the time a patient has mild to moderate dementia, the amyloid has already done its damage and triggered a self-perpetuating cycle of inflammation and tau tangles that amyloid removal cannot stop. This trial will help answer those critics. If we see a robust slowing of decline in the ADAS-Cog scores, it validates the sink hypothesis. If we see no change, it suggests that perhaps we need to intervene even earlier, or that albumin alone is not enough. No discussion of a clinical trial is complete with a rigorous look at safety and ethics. Safety concerns. 1. Fluid overload. As mentioned, albumin is an osmotic agent. It draws water from tissues into the blood vessels. For an elderly patient with a weak heart, congestive heart failure, a sudden influx of fluid volume can be dangerous.
The protocol likely involves slow infusion rates and careful monitoring of blood pressure and heart function. 2. Immunogenicity. Since this is recombinant, made in yeast or bacteria, the body might recognize it as foreign compared to natural human albumin. This could lead to antibody formation against the drug, rendering it ineffective or causing allergic reactions. 3. Coagulation. Albumin plays a role in blood clotting balance. Manipulating high doses could theoretically impact bleeding or clotting risks, though albumin is generally considered safe in this regard. Ethical considerations. The most significant ethical discussion here revolves around the open-label design. In a disease as devastating as Alzheimer's, the placebo effect is powerful. Patients and caregivers desperately want to see improvement. Without a blinded control group, people who think they are getting the drug but aren't, it is very difficult to know if a slight improvement in mood or behavior is due to the drug or simply due to the hope and extra attention the patient is receiving from the study staff. However, giving a placebo infusion to an Alzheimer's patient is also ethically complex. It involves subjecting a vulnerable person to IV lines and hospital visits for no potential benefit. The exploratory nature of this trial justifies the open-label approach for now, but any future Phase 3 trial will undoubtedly need to be double-blinded to prove efficacy to the FDA.
Informed consent. The trial requires signed informed consent, but in a population with dementia, this is tricky. The patient must have the capacity to understand, or a legally authorized representative, like a spouse or child, must sign for them. The study explicitly excludes patients who cannot perform the cognitive tests, ensuring that the data gathered is reliable, but also potentially excluding those who are progressing rapidly. As we close this deep dive into this trial, we are left with a picture of a scientific community that is relentless. Despite decades of setbacks, despite the billions of dollars lost on failed trials, researchers are not giving up. They are innovating. They are moving from brain surgery to blood infusions, from donor dependence to lab-grown abundance. This trial of recombinant human serum albumin represents a fusion of biotechnology and classical physiology. It asks a simple but profound question. Can we clean the mind by cleaning the blood?
We will be monitoring the results of this trial closely. The estimated completion is late 2025. When that data drops, we will be here to analyze it, to separate the hype from the hard numbers, and to explain what it means for you and your loved ones. Thank you for joining me on this journey into the cutting edge of clinical science. If you found this deep dive valuable, if you appreciate the detailed breakdown of the how and the why behind the headlines, please support the channel. Until next time, stay curious, stay informed, and keep asking questions. This is Clinical Trials Explained, signing off.